So, okay, recording in progress, excellent. Sorry, 1 second, back to slideshow. So, as Sandra very kindly said, I am a developmental geneticist getting into plant evolution, and I work with ferns. The reason I do that is because I want to understand how the world changed from an environment which was dominated by the ferns, like in this top picture, to a world where we were dominated by the flowering plants. So we are most familiar with flowering plants, and we think of them as plants because they are the dominant group now on the planet, but actually they belong to a much larger family. So these are called the land plants collectively. The flowering plants represent one small group, or most recently evolved group, within what we call the vascular plants, and we have the more basal bryophytes as well. And so all of these land plants have evolved from an ancestor within the algae. And just to explain that, we think that the original algal ancestor of the land plants had a life cycle that was almost entirely composed of a haploid organism. The only time it produced a diploid cell was when it actually mated and a sperm met an egg, and you got this single diploid cell called a zygote forming. That immediately underwent meiosis and recreated haploid cells to start the life cycle again. What makes the land plants different is in their last common ancestor, rather than undergoing meiosis, that diploid cell started to divide and stay diploid, and we got a multicellular diploid phase. So this is what we call the embryo, and when it gets larger, we call that the sporophytes. Sorry, plant evolution has a lot of technical terms like this, I'm afraid. We want to understand the genetics of how plants have evolved, and I should say that the history of plant evolution has essentially been the history of how that diploid cell multiplied and became more and more elaborate as an organism. So we use genetic models to understand this at the molecular level. And for those who aren't plant scientists, most of our models fall within the flowering plants. And in particular, we have Arabidopsis thaliana and rice as the two most famous. Outside of the flowering plants, we have a much more sparse situation. And in fact, until recently, we only had genetic models down in the bryophytes. We now have an awful lot of genome data, but if we want to understand the function of genes, we have to be able to change them inside the plant. And for that, you need model organisms where you can do genetic engineering. So in the bryophytes, we have two. So a liverwort called Mercantia polymorpha and a moss now called Physcomitrium patens. So in recent years, I have been involved in helping to develop a new model organism within this group in the middle here. So this is a more basal vascular plant than the flowering plants, and this fern we work on is called Ceratopteris richardii. So this is really good for me because, as Sandra said, I'm really interested in the evolution of plant reproduction, and it's only relatively quite recently that plants evolved what we think of as normal plant reproduction, which is reproducing by seeds and flowers. So it occurred in this lineage here, which comprises the gymnosperms, which is the conifers, and the flowering plants. So the ferns fall into the seedless category of land plants over here, and they actually form the most recent outgroup of plants that are still alive before plants evolve seeds. So the aim is that we want to use this and its genetics as a comparison against the seed-bearing system to understand what genetic changes happened here to actually led to the origin of seeds. But this is not the only thing that I think ferns and model ferns will be useful for plant evolution. So if I can give you another quick example. So as I said earlier, so we had the evolution of this diploid multicellular stage at the base of the land plants. In the bryophytes, this is an incredibly simple organism, and in fact, it's a simple structure. So this, what I'm highlighting here, that is the entirety of that diploid sporophyte in the moss Physcomitrium. So this, evolutionarily, is the equivalent to an entire Arabidopsis plant. So as you can imagine, trying to compare genetics between those two is very difficult. And I'll give you an example of that a bit later. So what we think happened at the base of the vascular plants is that diploid stage of the life cycle developed the ability to acquire indeterminate growth. And this is the evolutionary origin of the plant shoot. So you might think, well, we've already got plant shoots in the flowering plants. We can use those to understand them. We don't need anything else. But actually, along this lineage, there have been a series of developmental changes to that shoot mechanism, and the ferns represent a much more ancient way of working on that. So the easiest way to describe that to you is to actually show you how a fern develops. So I'm now going to want to take you through the life cycle of the fern that I work with, Ceratopteris richardii. I'm going to start at the point where this plant looks very recognizable to us. So when it starts out, it has a shoot, it makes leaf-like organs, and it makes roots. So you'd think, okay, that's a perfectly ordinary plant. However, when you get into the detail, we find significant differences. So at the top here, this is a diagram of what a shoot apex looks like in the flowering plants. So this is a dome of stem cells at the very top of the plant. As they divide and are pushed out, they leave a zone which is telling them to be stem cells, and they become co-opted into the production of organs. Now, this is a system that is entirely based on positional signals and communication between the cells. In the ferns, we find a very different situation, which is summarized in this picture here. So rather than having a pool of stem cells, you have a single cell acting as the meristem apical cell. That keeps dividing, it regenerates itself and produces daughter cells. And each of those daughter cells acts as a unit of development. It divides, and all of the offspring from that daughter cell form a unit like this. And from that, you get the specification of one of these leaf-like organs, a frond, and also a root. And both of those develop by having their own new apical cells specified. So this is a very different system from what we know in the flowering plants. And how we got from a system like this to the flowering plant's shoot apical meristem, we don't know yet. And I'm afraid from there, ferns only get stranger. So as the shoot ages, the fronds that it produces get more and more complicated. And that is because those apical cells persist for longer and longer. And you start getting more and more lateral apical cells being specified as well to develop these complex forms here. And in fact, from a developmental perspective, these things are much more shoot-like than they are leaf-like. And there is still a big debate going on about whether fern fronds are the same as seed plant leaves, or whether we have two different independent evolutionary origins here. So I'm not going to refer to these as leaves, these are fronds until we know better. And eventually you get this type of frond being produced, and the plant starts to look like this, which is very different from what we would expect from a flowering plant. But this plant has undergone a reproductive transition at this point, going from this type of frond to this type of frond. This is equivalent to flowering, but we cannot say yet that it is the same as the flowering transition in flowering plants, because we don't understand the mechanisms behind it. What happens is, each of these pinnae is a leaf lamina, and on the underside of that, you get sporangia being specified. And this is where meiosis happens. So you're getting the creation of haploid cells. In the flowering plants, this would happen in flowers, and meiosis would lead to the production of two multicellular haploid organisms, the pollen grains and also the egg sac inside developing seeds. In ferns, we don't get that. Each of the products of meiosis in here is a single cell that gets wrapped in a protective coat, and it's called a spore. And these are then dispersed into the environment. And then those germinate, and they form a totally separate free-living organism. So it also grows by apical cells dividing at the tip, but it becomes fully photosynthetic and free-living. And then this is what produces the sexual reproductive phase of the life cycle. So this is what makes eggs and sperm. So rather than having anything like pollination happening, in order to mate, this organism releases sperm into the environment, and that sperm has to actively swim and find an egg, which is held in an egg chamber on the gametophyte phase here. So once egg and sperm fuse, you then get an embryo developing on top of that parent gametophyte. This is referred to as a naked embryo, but that is a misnomer because it actually starts life inside an egg chamber. So you can see that compared to flowering plants, this is actually a lot of different things are happening here. And we at the moment understand precisely zero about all of it. So there is a lot of potential for people to investigate this and find out what's going on. And as I say, we use this particular fern, Ceratopteris, to do that. So Ceratopteris is a relatively good model fern, I think. It is quite small. And so here you can see my cabinet of science where I'm growing many ferns in a dense population. It has a 6-month generation time. So for those of you who work on plants, this is more equivalent to working on rice than it is to working on Arabidopsis, but that is still perfectly feasible. Most importantly of all, it can be genetically transformed. So we can actually change the gene functions inside it. And helpfully to that, it behaves genetically as a diploid organism. And this is unlike many ferns where you've had a huge amount of gene replication going on. And perhaps most importantly of all, we now have a genome available for it. Hooray, I've worked in the time prior to that, and believe me, it was difficult. So I want to give you 2 examples of projects that I've worked on using Ceratopteris to give you an idea of what we're able to do with this model organism at the moment. And so my first example is involving plant stomata, which I have to say is a direction I never thought my career would go in, but I took the opportunity when it arose. So as a quick introduction, stomata are the air pores that you find on leaves, and these control gases coming into the plants and the loss of water out of the plant. So people tend to think of the stomata as the hole in the middle, but actually what makes them work are the 2 guard cells that surround that hole. So these are actively and continuously sensing their environment. And when they receive the right signals, they will change to close or open that pore in the leaf to adjust the aperture and control gas exchange and water loss. And that is an active process. And one of the main signals that triggers the stomata to close is a drop in humidity. So we now know quite a lot about how that mechanism works. I am not going to bore you with the details. I just want to point out that we know of 4 general components to that. So we have signal transduction by a plant hormone that's called ABA. And inside the guard cells, you have signaling by reactive oxygen species, which is called ROS. We also have signaling from cytoplasmic calcium inside the cell. And these act on channels in the guard cell membrane that control ion movement. And so what happens is when the plant receives a signal, the guard cells open these channels, ions leave the cell. That changes the osmotic potential inside the cell. And so the water inside the cells then follows the ions in the separate water channels. That causes a loss of turgor pressure inside the cells. They deflate a bit. And so the gap between them shrinks and the stomata changes, reduces its shape. So simple when you know how. So this is all information that we've gathered using this model flowering plant, Arabidopsis thaliana. But this has left a very big question, which is, do stomata have active closure outside of the flowering plants? Because we know that all land plant lineages, with the exception of the liverworts, have stomata on them. We also know that in these basal bryophyte lineages, they seem to have a different function. So again, here we have that diploid sporophyte spore capsule from the moss Physcomitrium. This is the part of the moss that actually has stomata. So down at the base of that, which are highlighted in green GFP here. Their function in this spore capsule seems to be to help it to dry out so that it can then release the spores inside. So we now know that in molecular terms, Physcomitrium has a number of the components involved in active stomatal closure, but we are still unsure whether or not bryophytes actually use active closure or not. So people have turned their attention to looking further up in the vascular plants in these lineages. And when we get to the ferns, we have wound up with a real conflict because multiple different labs have looked at this question: do fern stomata have active closure or not? And basically, every lab that looks in a different species comes up with a different answer. Some say that there is active closure, some say there is evidence that it's only passive closure. And of course, there is nothing that a scientist hates more than another scientist turning around and saying, no, you are wrong. So this conflict has been raging in the literature for about 15 years. And one of the researchers involved in this came to me and said, could we have a look in the fern that you work with? And perhaps foolishly, I said, yes, I'm happy to do that. So first off, Ceratopteris does have stomata on the underside of its fronds, which is good. So you can see them here. The complicating factor is that Ceratopteris is a semi-aquatic fern. It likes to live in a puddle. So we grow this plant at 90% constant humidity. So these plants have absolutely no reason to try and control their water loss. So what we observed is that the stomata always seemed to be open. And so we weren't sure whether there was going to be a stomata response. And sure enough, if you take those plants out of their nice humid environment and just put them at ambient humidity, which is much lower, over the course of 2 hours, they steadily collapse and die. They have absolutely no tolerance to low humidity. But then we had a bit of a thought and said, well, okay, but they've never ever had to use their stomata. And we know that plants have the ability to acclimate to different environments. They can adjust their developments and their mechanisms to suit their current environment. So what we decided to try is to see is, could we acclimate Ceratopteris to lower humidity, and would that activate their stomata? So we did this applying a very simple pre-treatment where we basically just exposed these plants to very short bursts of low humidity as they were growing. And then when we repeated this experiment and took them out and put them at low humidity for 2 hours, it turned out they were much better at surviving that low humidity, which is very encouraging. And then when we actually measured the stomata area as a way of testing for response, so on the left here, we have stomata from these wet pre-treated plants. We saw no significant statistical response there over 2 hours. But in these dry pre-treated plants, what we saw is that the stomata did respond significantly in closed. So this was really exciting because we just shown, okay, yep, ferns can, well, Ceratopteris can close its stomata if it wants to, but this is an ability that ferns seem to be able to turn on and off. It's not constantly on, not constantly off. So that turns out ferns are actually cleverer than we thought they were going to be. But also this is something that might explain all of the contradictory results that people have been getting because everyone had been growing their ferns in a different way. So this was really quite exciting. And as a further test, we discovered that we could replace that drying pre-treatment by giving them treatments with this plant hormone ABA. So keeping the plants at high humidity, we would give them drops of ABA solution instead of exposing to low humidity. And when we repeated this experiment, we found that that was able to trigger them to close their stomata as well, rather than needing that dry pre-treatment. So you can see here, the plants are more tolerant. And then again, when we look at the stomatal aperture, under the mock treatment, there was no real significant change over 2 hours, whereas plants pre-treated with ABA, their stomata responded very quickly. So this was exciting because we knew that this hormone was involved with stomata in the flowering plants, and so this pointed to it using a similar mechanism. However, of course, this isn't very precise, and so we wanted to understand more. And so this was very much a side project for everyone concerned, and we didn't really have a budget for it. So we wanted to find a way of getting the most information we could as cheaply as we could. And to do that, we turned to RNA-seq. So what we had was a series of treatments that gave different responses. So on the left here, we have our untreated insensitive stomata, and then we had 2 different ways of treating the plants to induce sensitivity in the stomata. And then with those, we could put them either under low humidity or keep them at high humidity, and this would trigger different stomatal responses. So they either closed or they wouldn't close. So what we decided to do is to use these different conditions and do comparisons between them and look for changes in gene expression that correlated either with the acquisition of sensitivity or correlated with stomatal closure. So this here is a diagram of how we did that. So compared our insensitive to the dry sensitized and insensitive to the ABA sensitized. And essentially any genes that change expression under these two treatments similarly between the two, we decided that those would be good candidates for triggering sensitization. And similarly for stomatal closure, we compared conditions where stomata either closed or did not close, identified the differences in gene expression, and then compared those to see what gene changes were always associated with stomatal closure. And so this was to identify a closure signature. So we could have done this by isolating stomatal guard cells, but frankly, life is far too short for that. So we went for a much easier option, and we were using RNA extracted from entire fronds. So there would be hundreds of stomata on a frond like this. So I mentioned that we now have a genome for Ceratopteris. We did not have one available at the time we did this experiment. So our RNA-seq assembly was done de novo with no reference genome. So people were a bit suspicious. And what we have on the right here is a method of quality control for that. This is something called a BUSCO analysis. And essentially, this is looking at the transcripts we generated that were protein coding and using an estimate from that of how complete we think our assembly is, based on the gene families that we would expect to see from all of the flowering plants with published genomes. And so in the ring here, that is our Ceratopteris dataset. And I hope you can see that it compares very favorably to all of these other different sets, which are all what you get using this method from published genomes, which should give you the complete complement of all proteins. And in fact, it does a lot better than the original Ceratopteris genome that was published about a year later, so which I'm pointing to here now. I should point out that this was version 1 of that assembled genome. In the last year, there has been a reassembly and a reannotation, and version 2.1 is much better. I am now happy to use that for my own experiments. So that is a good advance. But this told us that the data set we gathered was fairly complete. So what did we find? I will take stomatal closure first. So we found 877 candidate transcripts that seem to be associated with the act of stomata closing. So that's a lot to look through, but of those, 51 were related to genes in Arabidopsis that we knew had a role in stomata. So we decided to use that as the basis for our analysis. So we took the most important components of the Arabidopsis stomata pathway, those genes. We looked for, relative to those in our assembly, whether or not they were associated with stomata in Ceratopteris, just everything that was available. And we've now compared that to our 51 transcripts that we think are connected with stomata in Ceratopteris. So this is what this diagram here shows. So the yellow circle in the middle, that's our 51 fern stomata closure transcripts. And the petals around it, each of those represents one of these gene families connected to an Arabidopsis stomatal regulator. So for example, this influx channel, we had 10 different genes in the Ceratopteris assembly. And you can see that one of them actually belonged to that stomatal regulatory candidate list, which is great. And of our 4 different signaling parts, so ABA, ROS, calcium, and the membrane channels, we found at least a couple of representatives of each within those identified closure candidates. So this makes us think that there is at least some conservation in the closure mechanism between ferns and Arabidopsis. And so that's exciting because we can now say that actually plant stomata had an active closure mechanism before flowering plants first evolved. It may have changed a bit, but it was there, and we've pushed that back into evolutionary history. Sensitivity is actually more interesting because we know far less about it. And the good news is that in our 2 different comparisons, we found 47 transcripts that were common between the 2. So these are very strong candidates for regulating stomatal sensitivity. And when we knew these were differentially expressed. And when we compared the direction in which they changed expression in response to being sensitized, between those 2 treatments, each of those genes went the same way. So 11 were upregulated in both the dry pretreatment and the ABA pretreatment, and 36 were downregulated. So this was a really robust signature and looked like really strong candidates. Of course, most of them found we didn't have a clue what they might be doing, but we found 3 from which had relatives in Arabidopsis that we knew were involved in stomatal regulation. So these 3 genes are MRP4, PDR12, GRDP1. So PDR12 is involved in ABA transport between the guard cells. GRDP1 is involved in fine-tuning the stomatal responses to ABA. And MRP4 is actually involved in preventing the stomata from opening again. So we think that what is happening here is that sensitization is causing the stomata to become more responsive, using the same mechanism as closure, but just putting it on a hair trigger. And it's possibly doing that by making it more sensitive to internal ABA signals. So this was really quite an exciting piece of work. And I hope I've demonstrated that Ceratopteris, even just using wild type, can be interrogated using techniques like RNA-seq, and we can get valuable information about molecular evolution out of it. Now, of course, all we've done here really is identify candidates. There is no validation with functional analysis. So I will now want to give you an example of a project where we have used functional gene analysis in Ceratopteris to understand a bit more about the evolution of a gene. And in this case, it's about a gene called LEAFY. So I'm switching gears. This is plant reproduction. So LEAFY is known as the master regulator of flowering and flower development in Arabidopsis. This is a gene that is not expressed during vegetative development. So the shoot apex starts its life producing leaves on its margins. And when the correct inductive signals are received to tell it to start flowering, it changes its identity to what's called an inflorescence meristem, where instead of leaves, it starts to produce flowers on its margins instead. And what we know is that in response to those inductive signals, LEAFY is turned on in that apex and causes that change to an inflorescence meristem. But LEAFY is also maintained in those side organs that become flowers. So flowers develop from their own meristem, called the floral meristem. And LEAFY is expressed in that and is necessary to make those side meristems produce floral organs. If you take away LEAFY, you get these leaf-like structures instead. And so the gene is named after the effect that it has. So this is where the idea that LEAFY promotes floral identity comes from. I will caveat here that in other flowering plants, there are variations on this theme where LEAFY's role is a bit broader. So it shouldn't be taken that this is all that LEAFY does in the flowering plants. This is all LEAFY does in our model Arabidopsis. But of course, this is all about flowers, and plants outside the flowering plants have LEAFY. It's found in all land plant lineages, but they don't have flowers. So the question rises, what was LEAFY doing before we had flowers evolve? And until recently, the only information we actually had about LEAFY function outside of the flowering plants was in this moss, this Bryophyte piscomitrium, which again, this spore capsule is the equivalent of the whole plant, effectively, for Arabidopsis. So the comparison is quite challenging, really. So people working on this found that the moss LEAFY, that was expressed throughout this diploid from the point where it was fertilized. And so here on the right, you can see a GUS reporter showing you that as it develops. And loss of LEAFY function disrupted development of that sporophyte completely. It failed. The trouble is, this is so different from anything that we see in our point of comparison, Arabidopsis. It's very hard to interpret what this is, what the effect here is, because this is a reproductive structure, but it's developing directly from an embryo. So is the failure a failure of embryo development, or is it just that without LEAFY there, you did not get the reproductive gene network switching on, and so it went wrong? So they couldn't really interpret what was going on. And to confuse matters further, they also found that LEAFY was also expressed in this other phase of the life cycle that moss has. So this haploid phase, which is much bigger and effectively forms the main body of the moss. So they found that LEAFY was being expressed in shoot apical cells at the top of these shoot-like structures, and also in these egg-producing structures called archegonia. So that suggested that there was an ancestral role here, which may, again, might be vegetative, might be reproductive. But when you knocked out LEAFY in this system, there was no actual effect on the phenotype. So no one was really quite sure what it was doing. So the interpretation kind of stopped at this point and everyone gave up a little bit. What they could say is that if you took a copy of LEAFY from Fiscomitrium and you tried to complement the Arabidopsis loss of function mutant, there was no complementation. So there has been a change in LEAFY function between moss and Arabidopsis. Perhaps not surprisingly, because they are separated by 540 million years of evolution. And when this was investigated further, they found that in the bryophytes, there has actually been, between the bryophytes and the vascular plants, there has actually been a change in the binding target that LEAFY goes for, changing that sequence. So it is presumably binding to different targets in most of the bryophytes than it is in the vascular plants. So this is perhaps not the most useful comparison about the evolution of LEAFY function. So what about inside these vascular plants? And there they found that there was the same binding target throughout the vascular plants, which was encouraging. But even then, if you took the Ceratopteris LEAFY and you tried to complement the Arabidopsis loss of function mutant, you only got partial complementation. And even if you took a gymnosperm sequence, you got slightly more complementation, but it's still not perfect. So this suggests that throughout the vascular plants, LEAFY function has been continually changing. So this made us really keen to understand what might be the function of LEAFY in Ceratopteris, as an example of that ancestral function in basal vascular plants. So for this, we had to do functional genetic analysis. So Ceratopteris is transformed by a very old school method that's called microparticle bombardment. It uses a bit of kit like this, which is a gene gun. So what we did is we knocked down LEAFY expression in the plants using a technique called RNA interference. So we essentially create double-stranded RNA in the plant that matches LEAFY gene sequence. The plant picks up on this and mistakes it for a virus, and it then goes and shuts down that gene. And to get a better idea of the expression pattern of Ceratopteris LEAFY, we also introduced a GUS reporter construct using a LEAFY promoter. And again, this is before the era of the genome, so I spent 6 months isolating that promoter by PCR. So what did we find? So we found that if we take away LEAFY function, that sporophyte shoot fails. So on the left here, we have an example of what it should look like at 25 days. And these three here are examples of RNAi lines that we generated. And so these shoots are really sick. They're just about producing a few fronds, but then the apex stops. And when we used qPCR to look at the expression level of LEAFY, of two different LEAFY genes in Ceratopteris, we found that in these examples, both genes had been severely knocked down. So loss of LEAFY function prevented this shoot apex from functioning. And in support of that, when we looked at our GUS reporter line, what we found is that LEAFY is expressed in that shoot apex, so in these points here, and not just in a situation where these plants are producing reproductive structures, it's expressed in the apex throughout the sporophyte life cycle, and that is including back to the point where it is only just finished being an embryo and that shoot apex is first starting up. So this is a clear difference from Arabidopsis, where LEAFY is only expressed when that apex becomes reproductive. And similar to moss, we also found a function for LEAFY in the gametophyte phase. So as I mentioned at the start, a haploid spore will germinate and produce a thallus structure very much like this after about 10 days or so. So this is our wild type example. In our RNAi lines, my colleague Steph Conway had real trouble establishing these lines because she kept saying that the gametophytes just aren't developing. And when we looked, we were finding examples like this. And so to my experienced eye, these look like examples of development just after germination, about 3 or 4 days. But these are 17, 13, 17, 10 days old. So these have arrested their development. Now, we did not see any GUS expression in our reporter line for this stage, but we would essentially would be looking for expression in a single cell because these develop from a single apical cell. So what my colleague Steph very bravely did is she turned to this technique called in situ hybridization. So looking for RNA expression directly in the tissues. And so she used two different probes to look for LEAFY expression. And what we're seeing here in recently germinated spores are this darker area indicates that there is LEAFY RNA present in these apical cells and the couple of cells just surrounding it. And when Steph also looked in our RNAi lines, she found no evidence for that. So this is strongly suggesting that, yep, Ceratopteris LEAFY is expressed in those gametophyte apical cells, similar to what we see in Piscomitrium. And then that is lost in our RNAi lines, consistent with a shutdown of apical cell function that we see in terms of development. And that's really interesting because when we put this back into our evolutionary framework, what we find then is that we can say, okay, in the common ancestor of ferns and what went on to be the seed plants, we think that LEAFY had a much more general role in apical maintenance. So both the vegetative and reproductive phase, and also both the sporophyte and gametophyte. And at some point during the evolution of flowers, so I should say that in developmental terms, we now think of flowers as a highly modified form of shoot. So it has its own apex, it produces its lateral organs. It's just been converted to a reproductive function rather than producing leaves. So you can see how that flower might have evolved from a shoot. LEAFY function may have been pre-existing in that, just as general shoot apex maintenance. And at some point, either during the evolution of flowers or just after, LEAFY function became restricted to just that reproductive context and was lost in the other vegetative shoot apices. So we can at least now get some explanation from that based on our work with Ceratopteris. So conclusions for that are just that, yep, we can do interesting functional analysis of genes with Ceratopteris. And using that, we were able to get some information about how this important regulator of flowering has evolved before the evolution of flowering. So as a final thing, I just want to very quickly outline where my group's research is going now. So as mentioned, we are interested in plant reproduction, and this transition from a stage where plants were reproducing without seeds, using this rather strange mechanism that ferns still have, and to reproducing with seeds. And this is something that I've taken to referring to as the seed revolution. Hopefully it will catch on, but if it doesn't, oh well. So we want to understand the genetic changes that are underpinning that. And we're taking two approaches. So the first is we are looking at candidate gene families that might have been involved in that. And the first of those are the Madsbox gene family. So two reasons for that. The first is that the Madsbox genes are heavily involved in floral development, and they are direct downstream targets of LEAFY. So they made a very logical point to start. And the second arises from a collaboration that sprang up once I moved to Birmingham. Some of my colleagues in earth sciences here asked me, would I help them interpret the development of a fossil that they had, this particular fossil species, Genomasperma. So this is a 3D reconstruction on the right. You might think this was a fossil flower from its shape and structure. No, this, they said, is one of the earliest examples of seeds in the fossil record. So they are confident that this part in the center is this gametophyte that is responsible for mating, and it's held on the sporophyte. And this surrounding structure is what they call a lobed integument. So this, they say, is what went on to make the seed coats in future evolutionary steps. And I have to say, I was jumping up and down at this point and going, that's a flower, that's a flower, that's a flower. So this is really quite exciting, I think, because from all those phylogenies I've been showing you, it's very easy to assume, yes, the seed came first and followed by the flower, and that they're two separate things. This kind of fossil is suggesting that actually seeds evolved from a much more flower-like structure than we might have originally thought. And so, seeds and flowers are more closely intertwined than we give them credit for. But also, because this looks so much like a flower, we can raise the hypothesis that actually the genes that now regulate flower development were originally regulating these proto-seeds and how they developed. And so again, one of the really hot candidates for that are these Mannsbox genes, because they control the identity of different floral organs. So that was a really exciting thing that I just did not expect to happen. And the second approach is a much more unbiased comparison, where we are not assuming certain genes are involved, and we're turning back to RNA-seq to try and understand how fern reproduction works at the genetic level, because at the moment we have no information about the genes that regulate that process. So I am at the moment in the process of gathering lots of RNA-seq experiments together to produce a data set that tells us these are the genes that are involved in fern seedless reproduction, and then we will compare them to our flowering plant models to see how similar they are, how different they are, and what that might tell us about evolution. So I think I've kept time, hopefully. Thank you very much for your attention. This work has all been done in collaboration with other researchers. So LEAFY evolution was done in collaboration with researchers at the University of Washington. Stomata RNA analysis was done in collaboration with Alistair Hetherington at the University of Bristol, and also researchers at the University of Oxford. This fossil reconstruction work was done here at the University of Birmingham in the lab of Professor Jason Hilton. So the LEAFY evolution work I've shown you was originally funded by the ERC on a grant held by Professor Langdale, for whom I was a postdoc. The Stomata RNA-seq analysis was funded by a grant given by the Gatsby Charitable Foundation. I am now being funded by the Royal Society, and I will be carrying on to try and understand the basis of the seed revolution with my two PhD students, Derry and Kirsty. Thank you very much for listening, and I'm happy to take any questions.
Thank you very much, Andy, for these very exciting talks.
You're welcome.
Sorry, yeah, it was very informative. So I don't know if you have questions yet. Please raise your hand or write it in the chat. Yeah, Pierre-Marc has a question, surprisingly, and then Matthias. Okay.
Good morning, Andy. Thank you for the great talk.
Oh, thank you.
I love hearing more about ferns. That's just amazing biology. I am really excited actually by the last bit of your talk on the let's say idea that potentially seed may have originated together with structures that look at least, you know, in terms of anatomy, much like a flower or something looking like a flower. So would you then even suggest that maybe in extant gymnosperms, what we have are species that are derived by losing these structures, and that flowers is actually just the evolution of this ancestral structure that diversified. And if you know, and that sounds a bit like a very aggressive hypothesis, but I don't know. And if you think that's possible, how would you test it with the different systems you have?
That is a very, very good question. So I think if we saying that seeds originally flowers is possibly too simplistic. I think what we always need to remember is that yes, plants evolve seeds, but there will have always been a seed-bearing structure, something that the seed is connected to. So I think in the very earliest examples, they are a side organ on a shoot. And there is, so in that fossil species I showed, Gymnosperma, that structure, which is the proto-seed, that grows out of something called a cupule, which has then been lost in the fossil record. So it's what I think probably what happened is what we now think of as seed identity and flower identity as two separate things. They were originally this one thing which held the gametophyte, and gradually over time they've diverged. So it's possible that things like gymnosperm cones, they are one descendant of that ancestral structure, which comprises the seed, the proto-seed, and the shoot that holds it, and flowers are the same, and that they've both elaborated that same underlying network in very different ways. Testing it, yeah, that's a very difficult one. I think really all we can do is we know a lot of the genes that are involved in things like seed development, and we just look at their homologs, orthologs in ferns and just see what processes they're involved in, because unfortunately, we are never going to get DNA out of those fossils, as far as I'm aware. And also, I mean, fossil interpretation is a bit of a tricky thing anyway. So I was sitting there going, yeah, but how do you know that that's the gametophyte? And they go, no, we've done lots of different anatomical studies. We think it's the closest. So there's always some uncertainty there, but at least by comparing living organisms, we might have a chance of looking at finding some hypotheses that will test that approach. And hopefully what we're going to be doing with the mouse box genes will start that off.
Thank you.
Okay, Matthias, next, thanks.
Yeah, hi, MD. Thanks for a really fascinating talk. So I got two questions. So the first one is, so LEAFY in Arabidopsis is then used by a switch in the photoperiod, right?
Yes.
LEAFY transcription. So have you tested if that's conserved in ferns? And my second question is related to the downstream targets of LEAFY, and with the genome being available now in ferns, if you look for the conservation of those targets.
Good questions. So my postdoc position came to an end just before that. So unfortunately, I did not